All right, we have just two more groups of macromolecules to walk through. First, let's start by taking a look at carbohydrates. Carbohydrates are going to be composed of simple sugars known as monosaccharides. Saccharides is the biological term for sugar, mono meaning one. So these are composed of one sugar. This can be glucose, fructose, deoxyribose, or ribose. Now take a look at these two on the bottom right, deoxyribose and ribose. What does deoxy mean? That's right. That's the removal of oxygen. Well, if you take a look down here at this carbon in the 2' position, what you'll find is that deoxyribose is short one oxygen from just plain old ribose. Pay attention to that. Now, polysaccharides are formed by dehydration synthesis when you actually bring multiple saccharides, monosaccharides, together. The formed bond is known as a glycosidic linkage. Now, there isn't going to be one particular enzyme that I can teach you here, but that's due to the fact that all of these different sugars have unique structures. And so you have to have a different enzyme which is capable of forming each one of these glycosidic linkages. But we can take a look into more detail at the different polysaccharides that can be formed. There are two major groupings. There's going to be storage polysaccharides. Storage polysaccharides store energy for later usage. And then structural polysaccharides, which are actually going to be a physical form, a strength and a support that is part of the cell. When we look at storage polysaccharides, the cell type matters a lot. For example, starch is found in plant cells. You'll notice that this is a single line of these glucose structures, and that glycogen is going to be the polysaccharide found in animals like you. That glycogen is highly branched. So if you were to count how many little carbon or little sugars are sitting at the ends of each one of those branches, you would notice that there are many individual sugars that we could kind of pick off of the little tree, right? Well, compare that to starches in plants. There's only 2 ends, so at most you could pick off 2 of those sugars at a time. Well, plants don't need quite as much energy as we do. For example, when you're running around your room, getting out of the house. These are all going to be things that require a lot of energy. Animals are very, very energy hungry. And so our storage polysaccharides have a structure that support that function. Structural polysaccharides, on the other hand, are going to all have very similar kinds of arrangements. Their base structure is going to be a chain of polysaccharides that form, and then hydrogen bonding links those chains together. For example, cellulose forms this cell wall for plants. And it's actually going to be the hydrogen bonds that link these chains together, which provides the strength for that cellulose to hold the cell together. Chitin is an example of a polysaccharide that forms the fungal cell wall. And then peptidoglycan is another example that forms the cell wall in bacterial cells. All of these are chains of sugars held together by hydrogen bonding, but it's the incredible number of hydrogen bonds that provide the strength in these structures. So tell me on Socrates, true or false, when eating a steak, you are eating your daily intake of starch. Hopefully you recognize this is false, since starch is going to be found in plant cells. But what if you had to name the type of carbohydrate that you are eating in your steak? Good, you're taking in glycogen. Now let's take a look at another example where these sugars actually form the backbone of another macromolecule. When we take a look at nucleic acids, the monomer of nucleic acids is known as a nucleotide. But nucleotides are going to be composed of 3 things. The first is a triphosphate tail. which you might recall we talked about when we looked at adenosine triphosphate. Adenosine triphosphate is actually a nucleotide. It just has some other functions in the cell as well, but its primary function is as a nucleotide, as a component for nucleic acids. Then attached to that triphosphate tail is a sugar core. For deoxyribonucleic acid, or DNA, it's going to be composed of that deoxyribose that we saw when we looked at the individual saccharides. If it's RNA, or ribonucleic acid, that's going to be a ribose core. And then extending off of that sugar is going to be a nitrogenous base. This is what determines the difference between the different nucleotides. So if you're a nucleotide of DNA, you can have an A, T, G, or C nitrogenous base. If you're a nucleotide of RNA, it would be an A, U, G, or C. The polymer of this particular group is known as a nucleic acid. So sugars are going to be that central component. As a reminder here, we have a 5-carbon sugar, which has a backbone of either deoxyribose or ribose. So this 5-carbon sugar, we can see here is carbon 1, here's carbon 2, carbon 3, carbon 4, and then right here is carbon 5. So each one of those little corners that's in that molecule is going to be one of those carbons. On carbohydrate, or on carbon number 2, is going to be that defining group that tells us if it's a ribose or a deoxyribose. On carbon 3 is a hydroxyl group, regardless of which sugar you have. And then it's actually going to be off of the 5' carbon that we attach our triphosphate tails. Off of this 1' carbon is going to be where we attach our nitrogenous base. Again, if it's an RNA molecule, then you would have a uracil. If it's a DNA molecule, you would have a thymidine. So DNA molecules are going to have 2 polynucleotide chains that are arranged anti-parallel to each other. Anti-parallel means that the 5' are going to be running in opposite directions of each other, and the 3' are going to be running in opposite directions of each other. So there, you'll see an example of this on the next slide. Deoxyribose is going to be the core sugar for each nucleotide, and then an adenine pairs with thymine when looking at that nitrogenous base pairing. RNA molecules are going to be single chains. They can fold back around again to interact with themselves, so they still have the ability to hydrogen bond to do this nitrogenous base pairing, but they're only going to be a single strand. They have a core of a ribose sugar, and their adenine is going to pair with a uracil. When we take a look at this core structure, you can see this anti-parallel nature with this strand down here being an example of a 5' end, which winds its way up, and this end here would thus be the 3'. Here is the 3' end of the other strand and the 5' end of that strand. This is anti-parallel to each other. And just like how you had to label the ends of your proteins, understanding that those are an N-terminus and a C-terminus, you'll always label the ends of your nucleic acids with a 3' or a 5' to indicate which carbon is at the end. Now this backbone is going to be a phosphodiester bond. It's the bond that forms between this 3' carbon and the phosphate attached to the 5' end. So this phosphodiester bond here is going to be a covalent bond. It's going to be a strong covalent bond. And so that backbone is going to be a very strong nature to it. By contrast, these nitrogenous bases are interacting with each other through hydrogen bonds that are known as base pairing. These hydrogen bonds are very specific. So you'll notice that G to C has 3 hydrogen bonds and A to T forms 2. So there's some specificity to it. And you'll also notice that even just with these 4 nucleotides here, there's many hydrogen bonds. So when you consider how many of the hydrogen bonds there are, there's actually quite a bit of strength holding these 2 nucleotides together. But it is definitely weaker to hold the 2 strands together via hydrogen bonding than the actual backbone nature of it itself. Now, if you were to take a look at the formation of this phosphodiester bond, again, here we have an interaction between this 3' hydroxyl group and this 5' phosphate group. This is going to be the formation that forms that phosphodiester bond, and it's going to be catalyzed by enzymes called polymerases. So I want you to take a look at this picture here and then tell me on Socrative. Pause if you need more time. Would this be an RNA polymerase or a DNA polymerase? Hopefully you identified that this is an RNA polymerase. And the reason you know that this is an RNA polymerase is because the 2' carbon here has a full hydroxyl group, which means that this core nucleotide is a ribose. We have a ribose sugar here. And so therefore, this has to be an RNA polymerase catalyzing this reaction. Now, the role of nucleic acids is something we are absolutely going to be expanding upon. And we are going to draw back to the structure of nucleic acids in the 3rd unit of this class so that we can connect that as we start to do these functions. So please don't forget about these structures. You're going to see them again. But just to give you a basic introduction, DNA is responsible for forming that permanent copy of genetic information, which is stored inside of your nucleus. RNA's job is to be a copy of that genetic information that can leave the nucleus, and it can be used as a template for protein synthesis, or it can have other jobs and roles in regulating that expression. You'll notice here that there's a number of macromolecules that are present. We have a DNA molecule here inside the cytoplasm forming its core unit. We have an RNA copy of that. We have phospholipids here that are forming this barrier. We have a transmembrane protein that this RNA is using to leave the nucleus. And we have a protein here, which is the enzyme ribosome, which you guys are going to learn is made of both RNA and protein. And you have the fact that this RNA is serving as a template for the synthesis of this polypeptide chain. The central dogma is going to connect this whole process together. It's what you see here, the idea that DNA is going to be copied into RNA. This is a process known as transcription. And that this RNA is eventually going to be used in the translation of proteins or polypeptide chains. DNA to RNA to protein is the foundation of information, and how that information is not only stored in the cell, but actually becomes something that produces a structure that produces the function. And that's how your cells actually work. Now, we are going to be continuing this by building these macromolecules up into cellular components. So expect to see things like what we've just done here in the last 2 slides next class. And I'm going to be constantly asking you to go back and be thinking about the macromolecular components that are here and connecting them to these additional and future structures.